I'd like to welcome Jason Sylvan from Texas A&M Oceanography Department. I first met Jason as a PhD student, or possibly as an early postdoc, and now he's an associate professor, but really just ages me, I think. And anyway, he's kindly agreed to join us to talk about microbiology, and you know, throughout his career, he's sort of been at the forefront of developing microbiological approaches, utilizing scientific ocean drilling. Okay, thanks, Jason.
Cool, thanks, Damon. Yeah, so thanks for having me, and I'm going to talk today about, I guess, start off talking about the South Atlantic TRANSECT project, and with full acknowledgement up front that probably the first third of this talk I stole from Roz, so sorry about that, Roz. And then I also wanted to just touch, since this is a workshop thinking about writing proposals, and in particular for mission-specific platforms, just touch on some of the things one should consider if they're thinking about either including microbiology in their project or having it focused on microbiology. So, and if you don't know who I am, my name is Jason. I'm an associate professor here in the Department of Oceanography at Texas A&M University, which is also one of the sites of one of the core repositories for IODP. And I do want to acknowledge all of the people that put work into writing the proposal that led to this. So IODP expeditions 390 and 393 will be implemented kind of, two expeditions as one project. The co-chiefs are myself, Damon, Roz Coggin, and Gail Christensen. But, a lot of people put work into the proposal. You can see their names all here. And in particular, I just wanted to point out Brandy Reese, because we've she and I thought a lot about the microbiology. My expertise is more in basement, hers is more in sediment. And so it's been a nice collaboration working with her on this. The goal of, or I guess the broad view of expeditions 390 and 393 is to go out and sample a flow line of the mid-Atlantic ridge. I'll talk a little bit about this. Originally, I should be right now celebrating all of the fantastic successes with Roz that we had as we went out and did this last fall, but obviously that did not happen due to the pandemic. So things have been delayed a little bit, and scientifically, I think we've actually gained from that delay and have been able to do some engineering work up front. And the JOIDES is actually sitting in South Africa at the moment, waiting to go on a second engineering expedition, which allows us to focus hopefully just on drilling and collecting great samples when we go out. So currently, the plan is for Expedition 390 to leave 7th of April in 2022 and spend 2 months at sea, and then be followed immediately by Expedition 393, June to August. And so I think originally there was going to be 6 months between those two cruises. And I think one of the nice things that's come out of this unfortunate overall situation is that these cruises are now back to back. So scientists won't have to wait as long, scientists on the first party, for the sampling party, which is necessary for this type of expedition after the expedition, since there's 2 of them and people might want samples from both cruises. That's also, MSPs will typically have a sampling party too, so something to know about. Okay, so I was going to get rid of this slide and then realized how, if you are not aware, what a fantastic visual artist that Roz is. And so she designed all of these logos, and also they will pop up in the corners of some of the slides that I clearly stole from her. So I decided to leave this in. But South Atlantic TRANSECT, I'm going to give you a little bit of a background on the project. So this is based on a proposal that was titled a multidisciplinary IODP investigation along a crustal flow line across the western flank of the southern mid-Atlantic ridge, and with the 3 main hypotheses, or main, I guess, purposes, are to study the hydrothermal evolution of the aging ocean crust, to investigate sediment and basement hosted microbial community variation with increasing substrate age, so as that crust ages, and to look at the paleoceanographic evolution of the South Atlantic. I do want to point out the first two of these are kind of the primary. However, the paleoceanography work will also be important as well. And these part of our, I won't go into it too much here, but part of what we use to get people excited about this work is that this is near the DSTP3, like 3 sites, which was the expedition that confirmed plate tectonics. And so there's some kind of exciting history to build on with this project overall. One of the reasons I wanted to show you this slide is just to point out the interdisciplinary nature of the project. And I think, you know, we all have our own interests, but I think one of the things that really helped sell this project in particular, and other ones as well, is how kind of integrated the different disciplines were. And that's seen as a strong point by, I think, by the reviewers as well as everybody involved. This proposal, I also wanted to point out, was the result of 2 workshops. You know, I think it was, Roz remembers the history a little better. I remember going to both of these. I think we started talking about it at the Denver workshop in April of 2012, so 9 years ago almost. And then it was more kind of built upon specifically during this TRANSECT drilling during transits workshop, at which point there was consideration of essentially sampling as the ship goes from one site to another. The way the ship moves around has changed, so it doesn't do that anymore. However, the proposal came out of this, and there were a few things that I wanted to point out. You know, one is that, you know, as Damon mentioned, right, he saw me when I was just a little baby scientist. And so in November 2013, I had just started a research scientist position, hadn't even interviewed for Texas A&M yet. So it's kind of, you know, 2, I guess, career positions ago, I guess. Now I'm an associate professor here. And so I started with this pretty early in my career and would encourage other people to do so too, because even though the expeditions, I think they can be a lot of work, there's a lot of steps along the way that you can be involved in that aren't kind of overwhelming at the earlier stages of your career. And I would surmise that this project overall has helped my career advance, and it's something that I'm really proud of, but was, you know, not sure if it was the right decision at the time. So only in hindsight, I can tell you, I think that it was. And thinking back to that November 2013 workshop, I was like looking through some old photos and found this one. I was like, wow, that's awesome. This is like what we were talking about at the time, a more generalized kind of TRANSECT. This is before we had the proposal in its final form. But you can see that's the writing on the board from that particular workshop back in 2013. And it talks about, you know, South Atlantic and doing a TRANSECT, thinking about crustal age and heat flow, and how sediment thickness might influence processes of crustal aging and hydrothermal flow. And I just thought it was really cool that that's actually what we ended up, or what we will end up looking at, you know, I guess it'll be 10 years later when we finally go on this expedition. And then something that I've just always found amusing was this. So this is a picture of Debbie Thomas, who's currently the dean in our College of Geoscience, looking during that workshop at some sites that people were interested in for different reasons. And at the time, probably still, there were several sites that were not drillable because there might be pirates. Okay, but so getting back to the South Atlantic TRANSECT, the strategy that we ended up deciding on was to drill 6 sites along a crustal flow line at 30 degrees south on the mid-Atlantic ridge. The thing that I think is really super cool, and it makes for exciting science, is that all of these sites were formed on the same segment of the mid-Atlantic ridge. And so you're truly looking at aging of kind of the same piece of crust over time by drilling these 6 different sites. There's 5 different ages, ranging from 7 on the young side to 63 million years old on the older side. We picked 7 because it, it's younger and it kind of allows comparison to previous work at, I think, 1250-60. And we picked 63 because it's close to the 65 million year old hypothetical ceiling of ocean crust, even though we know that there can be circulation in older crust, but this lets us look at kind of all of the things that happen up to that age. And we picked two ages, two sites at the age 63 million years to look at differences in sediment load and how that might affect hydrothermal circulation. So, and we aim to recover complete sediment sections, and the uppermost basement below that sediment, kind of as far as we can go, between 200, 250 meters, given the allotted time. So this, the strategy exploits targets that can simultaneously address multiple IODP science plan themes to maximize the scientific output of the drilling effort. Again, just bringing us back to this idea that interdisciplinary work is powerful, and I would always argue that it creates good science. I think as a microbiologist, most of what we do in regards to IODP tends to almost have to be interdisciplinary to really understand the environment that the microbes are living in and say something interesting about what they're doing. But I think that these also review well. So our overall objectives touch on a few of the main objectives of IODP, the main themes, the Earth connections theme, which is one of our primary goals, quantify the timing, duration, and extent of ridge flank hydrothermal fluid rock exchange. In the biosphere frontiers theme, we will investigate sediment and basement hosted microbial community variation with substrate composition and age. And in the climate and ocean change theme, which is important, but one of the secondary themes, or the secondary theme, we'll investigate the responses of Atlantic Ocean circulation patterns and Earth's climate system to rapid climate change during the late Cretaceous and Cenozoic, which is the age of the sediments that we'll be recovering. So moving forward, I'm going to focus just on the biosphere aspects of this project, and what we thought was interesting here, and also some considerations for doing microbiology as part of an IODP expedition or project. So in this particular case, so one of the things that interested me to this project, in particular the South Atlantic, if you look, this is a map of obviously of the world, and showing in red, those are at the time, the paper is a few years old now, sites that were drilled by IODP, and in green, sites drilled during ODP, and the larger circles that are outlined in black, like this one, and that one, and so on, those are sites where microbiology is one of the primary goals of that expedition. So you can see, not surprisingly, but microbiology is kind of a late bloomer in the IODP and ODP world. And so most of the sites don't have that. There's only been a few expeditions that really focused on microbiology more recently, but the entire South Atlantic Ocean pretty much is a mystery as far as the type of work we do. And so this lets us ask a lot of big open-ended questions. One of the things that we wanted to look at, so this figure is from a really nice paper from 2012. On the x-axis, we have depth into sediments, and on the y-axis, cell concentration. And by looking at different sites all over the globe, Cal Meyer and colleagues were able to estimate the total number of cells in sediments below the seafloor, and also to show some generalized patterns where you have more near the surface and less with depth. And then depending where you are in the world, this yellow represents South Pacific Gyre open ocean sediments. It's actually these sites right here. And the black sites represent some old ODP sites that were closer to the shore and subcontinental shelf and places like that, where there's a lot more organic carbon input. And so you can see, depending on where you are, the number of cells you start off with in the surface can range up to 4 or 5 orders of magnitude. And if you put all this data together, you can make this kind of global estimate. However, it's a fantastic paper and really important, but none of the data in this figure comes from the South Atlantic. And so, this region, large region of the ocean where we have literally no data for microbiology. So part of it was just that, techniques have gotten a lot better. We can do a lot more nowadays, and we can go into this region of the ocean that's unexplored for the type of work we do, and ask kind of the who, what, when, where, and why questions. What bugs are there? How many of them are there? What are they doing? We can also think about questions related to limits of life, because it's far enough out in the middle of the ocean that the organic carbon content is pretty low. And definitely think about the significance, of microbes in kind of that area of the world to biomass as it relates to that type of figure, and also biogeochemical cycles. So I do want to point out that this figure, which I've reproduced that figure here, this is representative of just sediments. And so there's very little data, even just cell counts from basement. And so my lab has generated some of that data that's hopefully coming out in different papers. And we're working on kind of the same type of analysis based on the little data that we have to get a global estimate of how many cells are in basement versus sediment. However, you could see looking at that sediment figure, there's a lot of data points there. The basement is way less data points. And so, having 5 sites that we know, or 6 sites that we know we're drilling into basements and getting samples from will greatly increase our understanding of what's happening in basement, and in particular along in age TRANSECT, which is really exciting to me. So one of the other questions that we kind of are able to look at is that this shows a map of modeled organic carbon in weight percent at the seafloor. And in particular, North Pond, which was Expedition 336, and Nankai, which was Expedition 316, represents kind of 2 ends at Nankai and kind of higher weight percent total organic carbon. You have more, I guess, quote, typical sediments that go anoxic at some depth, not that great, or not that deep. And the typical geochemistry you would expect with that. North Pond is kind of a transition where you can go pretty deep with oxygen still in the sediments, sometimes the basement, sometimes not, depending on the site. And then South Pacific gyre represents, the lowest, most oligotropic type of environment where there's oxygen to basement. And so in the current area that we're looking to sample during the South Atlantic Transect, it's really intermediate between North Pond and Nankai. And so it lets us ask some interesting questions about, what that might look like. Will we run out of oxygen? And, at all of the sites, does it matter with age? Some really, really cool questions. So our plan at each site is to triple APC drill the sediments, put in a reentry cone, and then drill 200 to 250 meters in basement using the rotary core barrel RCB, and also do some wireline logging, which will help us understand the holes and in particular, the regions that are not recovered. And as I mentioned, because of the delays, we were actually able to do some engineering work up front. And so during when 390 was supposed to be out, instead 390C went out, Expedition 390C, which went out with no scientists, but did bring Emily Estes, who's the staff scientist for 390C, and they were able to drill 4 out of the 6 sites and start installing reentry cones. And so the great thing is this would have all taken time during Expedition 390. And so we now have that stuff complete. And hopefully when we go back out there, our chance for success is much higher. And then additionally, there's plans to be another follow-up engineering expedition that's actually waiting to leave Cape Town at the moment. So moving from kind of South Atlantic TRANSECT specific to just broader thinking about IODP microbiology, I wanted to just give some, I guess, some of my thoughts about things to consider if you're thinking about conducting or adding microbiology to your project. So, in particular, biomass can be really low in subsea floor environments. So clean sampling and also clean sampling and processing during post-cruise work is necessary. So, if you If you're leading one of these expeditions and aren't the microbiologist, there's some things you need to think about. If you are the microbiologist and you're going to have these samples back in your lab afterwards, there are some additional things to think about. So some really basic precautions like masks and gloves are needed while working with samples, including in the core splitting room. Nowadays wearing masks is pretty much the rule. And so I think it's less onerous for us, but it's really important with working with these types of samples. Despite that, I don't want to, I'm not here like raising alarm bells. It's just, these are things you should think about ahead of time. Good data from these environments, both sediments and rock cores, is possible despite the challenges. It just takes some kind of pre-planning. One other thing to think about, depending on how important you want microbiology to be in your particular project, is how many microbiologists are there. I would argue that 2 scientists are really mandatory just to cover the shifts, 24 hours. Otherwise, I've been the sole microbiologist on an expedition, and I got samples and did work, but part of it is I'm only good at some aspects of microbiology, and so you'll just increase the kind of different things one can do by bringing along 2 experts, and also just you can't cover 24-hour shifts with 1 person. So, and if you're looking for a project where microbiology is a large part of it, 3 or 4 microbiologists is much better. You'll be able to actually do experiments. With 2 scientists, you can essentially collect the samples and process them. With 3 to 4 scientists, you can start doing some experiments, shipboard. So, and I mentioned some of the precautions to think about. And so this is a picture from Expedition 360. Hello to Alessio, I saw you earlier, who was on that expedition as well, and showing Benoit Ildefons, who's a geologist, working with Jenny Edgecombe, a microbiologist, to pick out a sample. They're all wearing masks and gloves. People were really supportive of this. And Jenny, because she's working with the samples later, is also wearing a lab coat. In general, we actually usually sampled, so we would usually pick our samples, say something like this sized, or this, not a roller, but something 10 to 20 centimeters long. We usually pick those through the core liner and poured it into a bag. This picture was a little bit staged, but still shows you that you need to kind of wear proper things to keep the samples clean. Microbiology tends to take their samples first. This is the best way to do it. And then once we take our sample and go to the lab to start processing it, then everything else can kind of return to normal. But while we're collecting our samples, we need to think about that. So both the Jordy's Resolution and the CHICUC have labs set up to do microbiology work. And some advanced planning with the operator helps. So on Expedition 360, I helped set up this kind of clean room type environment where there's plexiglass walls and a curtain, a vinyl curtain that you go in, and the air comes in through the top. Through here, through a HEPA filter. So essentially it's an overpressured area where the air that's in it is filtered and goes out the bottom. And that, I think, worked pretty well. Since then, the JR bought something called a Coach, K-O-A-C-H, which is kind of like a traveling mini clean room. And they have these on the CHICU as well. It's a Japanese unit. And I point this one out in particular, because you can move it around, so that, the one catch with this version I use on 360 is you're kind of stuck in that one spot. But B, you can move it around, and this is a Yuki Morono on the JR using it, but he also went on Expedition 357, which was one of the MSP projects to Atlantis Massif, and brought this on the ship that did that drilling. And so this is the type of thing you could use on an MSP if you want to do microbiology and have a clean sampling environment, even if the ship wasn't kind of purpose-built for that. So I kind of mentioned here, special considerations are needed for MSPs since the equipment isn't standard. That said, in particular, Expeditions 357 and 364 are really great examples where this was successful. You just need to plan ahead. So just for scale, you can see there's me working in that kind of clean room environment in Expedition 360. So, and I think the last thing, or one of the last things I want to say is that the, microbiology in IODP plays well with others. So I think, we could do whatever fantastic microbiology we want to do, but the story is always more compelling when we include other disciplines. And so in this case, this was a paper that came out last year based on the work from Expedition 360. And on the right, you have some kind of fancy RNA-based analysis that tells us what microbes are doing, what types of metabolic things. And that's all lovely, even though it looks horrifying. But this was only really convincing because it was combined with some Raman analysis and also some work looking at lipids. And my lab did also had some biogeochemistry that's not in these figures. And so kind of all of that together, I think, is what made it for a compelling story. And so, the microbiology both fits well into proposals, and if it's going to be the lead, it plays well with the other disciplines. And then I will wrap up with mentions of just two things. So one, if you're considering doing microbiology on IODP expedition and haven't done that before, I'll be submitting this is like in its final stages, what will be a IODP technical note, which is a type of publication that IODP puts out. They're free and accessible from everywhere through the IODP publications, called Recommendations for Microbiological Sampling and Contamination Tracer Use Avoid for the Droides Resolution, blah, 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 following 20 years of research. This is resulting from a really small workshop with myself, Rick Caldwell, Beth Orcutt, Dave Smith, and also working with two of the staff scientists, Kara Bogus, who was a staff scientist, and Emily Estes, who is now. And so we have a lot of kind of recommendations about, what one can do with different numbers of saline microbiologists and how, some thoughts about contamination tracer and what works best. And so I would recommend you to have a look at it because we wrote it specifically for people to look at as they're kind of in the planning stages before they're on the ship. So, and then since this is a MSP specific workshop, I also wanted to point to these papers. So mostly just, this first paper is from Scientific Drilling and is specifically about using the MEBO and the British rock drill and kind of fitting them to be able to make contamination tracer analysis and inject the tracer and collect those samples. And then also just to show that these are two examples of 357 and the Chicxulub drilling that have resulted in really fantastic microbiology work. And so it is totally possible and just, requires some thought up front. So I will end here. I think I'm about at my time, but the South Atlantic TRANSECT is the thing I'm kind of thinking about most coming up in the future. It's a big interdisciplinary project looking at multiple aspects of what happens as the crust ages. And so I'll stop there and take any questions. Thanks for your time and listening.
Okay, thanks a lot, Jason. Really good. Yeah, I think the take-home message is that microbiology is completely possible from mission-specific platforms. It can add really valuable science, but it does need to be prepared for.
Yes. Yeah. I think with the MSPs kind of in particular, just because they're not standardized, right? A lot of the aspects of the drilling, right?
And just before this, we had some various groups presenting some ideas as sort of pre-proposals, and some of them are proposing approaches that are technically challenging but not impossible, I would say, and that there might need to be some development. And of course, there was a lot of development before the Mid-Atlantic Ridge 357 wireline drilling, using the rock drills. That was done on pretty short order in order to collect fluids from holes, to do contamination testing, and so all this is possible, and developments can happen.
Yeah, and in the example of 357, one of the things that I thought was interesting was that Beth Orcutt, who kind of led that part of the effort, the microbiology, wasn't on the original proposal, I don't believe, but was brought in because they knew that she could help kind of execute, in particular, that aspect of the work. And so it's something to consider. You might have to kind of reach out to people that know how to do the thing that you want to do, even if they're not on the proposal. It's probably good to bring them in at the proposal stage, but these things are possible.
Sure. Any other questions from the wider audience?
Sure. I see a question from Steve.
Yeah, thanks for the talk. Very nice. I was just wondering, for the South Atlantic TRANSECT, how have you negotiated, I guess, with the geologists, because you sort of need to take your sample, very quickly, and you might
Right.
not know exactly what you've recovered, and you might, for the geologist, you might be taking away something valuable as a whole round. How have you sort of managed that? And I guess with regards to MSPs, often the amount of material that is recovered is even lower than what's recovered on typical JR crew, so it becomes even more important.
Yeah, that's a super good question. So Roz and I decided on a Fight Club type approach, but because of what you've mentioned, that in particular, with sediment, I think it's probably less of a concern. With basement, right, recovery is often not great, and then there might be one particular section that's everybody's fighting over. So we based it on what I've done before and other people have done before, is that essentially when the cores come up, there's some kind of somebody assigned from the geology group that will go look at the cores with the microbiologist that's going to sample it and kind of make sure that you're not taking that one critical piece. I think there are times that there's conflict, and so depending on those two individuals, my opinion is that the best way forward is you essentially have to just kind of trade off taking the hit, right? One group can't always get the best samples, but I think that there needs to be kind of communication to make sure that you're not, what might be kind of a standard sample to one group, but a critical sample to another group doesn't get lost to that group that then can't do some cool science. But,
yeah, I mean, we can also, quickly photograph things. We can get a lot out of
oh,
photographing
yeah,
the outside of the core. And, it's very rare where things are truly unique. I mean, the other thing is that, the microbiologists tend to work on the interior of the core, not the exterior of the core. So there is quite a lot of material that, although it gets dismembered, it can be returned for petrographic or geochemical analysis if we're careful and keep track of it.
Yeah, so we'll often kind of split it open and try to sample. The things we fight the most over are probably veins, but we'll split it open and bring back kind of the external parts of the core, because for us, that's going to be too contaminated to work with anyway. And as Damon said, we always take pictures kind of from 4 sides to have at the very least that photographic record.
Did you take that into account at all when you were deciding how much you want to drill? Did you increase the amount that the depth of any of your holes to try and get more material so that you can share it, or did it have any effect on that?
Roz can correct me, but I don't think so. I mean, because on average, for every 10 meters that comes up, or 10 meters that's drilled, I shouldn't say that comes up, we usually take 1 to 2 10 to 20 centimeter pieces. So, it's over the course of the whole core, it's like 1% or less of the actual rock. I think the challenge that can be frustrating is it's a destructive sampling, right? So that piece is gone, but it's a tiny fraction. So in terms of thinking about the whole portion, more is always great, but it wasn't.
Yeah, I mean, I'd say we didn't explicitly do that, but there have been discussions of some legs where you would drill a specific hole for microbiology. And
just to follow on from that, we didn't do that for the hard rock, because making a hard rock hole is hard enough, and we're just going to go as deep as possible. But we do deliberately triple APCing the sediment so that we can have a continuous spliced record for paleoceanography, but effectively take lots of samples from the third hole, as it were, for geochemistry as well as microbiology. So there are other samplings that require whole round a single APC.
Okay, thank you very much, Jason.